Hi everyone, this is the HPRC Epigenome browser, a portal built by the Wang Lab at Washington University in St. Louis for exploring epigenomics data from the Human Pangenome Reference Consortium, HPRC. In the next few minutes, I will take you through the whole thing, picking samples, configuring tracks, visualizing them in the genome browser, and saving your work. In the end, finding out what data actually exists. Let's go. The data consists of 232 individuals with haplotype resolved genome assemblies, drawing from 28 subpopulations across 5 continental groups, with about 12 terabytes of epigenomics data on top of them. DNA methylation, gene expression, chromatin accessibility, and chromatin conformation. The portal is organized as 7 tabs, and the first 3 are the workflow: sample, track, and browser. Pick what you want, decide how it's displayed, then look at it. Everything else, session, data availability, tutorial, and about, supports the first 3. Up in the header, there is a reset button that clears every selection. And the day-night toggle that applies across the whole portal. The sample tab is consists of 4 steps, and each one stays highlighted until you've answered it. So the highlight works down the page as you work. Step 2. Choosing the reference genome. 2 choices, HG38 and T2T CHM13. Step 2. Picking up a sample. You can search by ID. Or filtered by the superpopulation. Use drop-down box here. And click again, you can remove a sample. Under each sample ID, you get a population badge, a sex badge, and a row of small color dots showing which assay that sample actually carries. Cyan for methylation, green for expression, orange for accessibility, purple for conformation. These colors mean the same thing everywhere in the portal. Step 3. The data layers. There are 4 cards: the methylation from ONT or PacBio, expression from IsoSeq, chromatin accessibility from FiberSeq, and chromatin conformation from HiSeq. Click to toggle. The coverage varies a lot, and we will show from the data availability later. The step 4 is the data overview of what you have selected. You can drag the rows to reorder, which controls the order they appear in the browser, remove with the X, or open the detail for the full metadata records. Scrolling down, and there is a map of where the samples are coming from. Each dot is a subpopulation, and the dot itself is a small chart. The core contour, color is a continental group, and the inner ring is a sex split, and the outer petals are 1 wedge per assay. The longer the petal, the more of that subpopulation has that data. Hover it for the full breakdown, and you can click to pin it underneath. Dots are nudged apart so they don't overlap with each other. And where one had to travel, an arrow points back to where it really belongs. For example, the STU here. Below that is a PCA plot. The 1000 genome in the background are in gray. The HPRC samples are colored by population, and everything you've selected is drawn larger. Scroll to zoom, drag to pan, and there is a reset button. Then we go to the track configurations. Your selection have expanded into individual tracks. There are 3 counters at the top: the total amount of tracks, and how many are switched on, and how many match your current filters. You can search across track names, for example, sample ID, track type, HAP1 or HAP2. You can select all, that means all the tracks here, or just select the filtered. You can deselect all or just deselect the filtered. Now we can just enable all the HAP1 tracks by first searching HAP1, then select the filtered. But to keep the tutorial session running, let's just select the default for the HG00261 by resetting and reselecting. Then next we go to the browser. You probably have to wait a little bit to let the browser load all the tracks. And this is a Washoe Epigenome browser running inside the portal with your tracks loaded. It opens on Chromosome 7 around 27 megabases. You can see the HOX-A clusters, which is a good region to see a lot of things. Now, the things that actually makes the portal different from a normal genome browser. Most of the epigenomic data here wasn't measured against the standard reference. It was aligned and processed against each donor's own assembly in that donor's own coordinate. So the portal ships a genome-aligned tracks that maps each haplotype back to the reference. For example, here is HG00621 haplotype 1 comparing against HG38. That's why you can put all the methylation from several genetically distinct donors side by side at the same locus and actually comparing them, even though the underlying sequence differs between them. The alignment tracks also show you the structural difference between themselves. You can show insertion, deletions, and inversions. Press the full screen for the full screen, and you can also use the shortcut F to toggle between full screen. The browser is simple to use. You can just click to move left or right and use the zoom function to zoom into a specific region. And for more tutorial, go to the Washoe Epigenome genome browser tutorial section. Now, if you found your work has been interesting so far, a session is a bookmark for your entire setup: the reference, genome, samples, data layers, which tracks are on, and where the browser was pointed at. The panel here shows exactly what would be captured right now. Let's save the current setup with the example name and save the session. Actually, we already have that one. Let's choose a different name. Next time, if you come here fresh, but you don't want to go through all the selection process, you can just click on the load to go back to exactly where you were left off. You can also export all the sessions you saved earlier and send it will be saved as a JSON file, so you can share with your colleague, and your colleague can import from here. Now let's move on to the data availability tab. This is the same map you saw in the sample tab. The data availability tab answers what actually exists. The track explorer can show what tracks are available. It can be shown by different track type, for example, genome align, methylation, expression, promoting accessibility, or you can search a specific sample to see what's there. You can download them individually or download the filtered tracks here using the download TSV button. Or instead, you can download the whole catalog using download full track list. There are 8,300 there, and more tracks will be added later. Here is the cohort and data coverage. Shows you what data are available here by superpopulation and subpopulation. And the matrix itself: 1 row per sample with population, superpopulation, sex, and the column per data type. The badge tells you which coordinate system the sample has data in. Blue for HE38, green for Cetram13, amber for DSA, which is the donor's own assembly. Every column can be sorted. The tutorial tab shows how to use the tab, how to use the browser. You can also start an interactive guide that gives you a quick introduction. That's all about HPRC Epigenome browser. If you encounter any issues, you can go to the GitHub issue tracker page and submit an issue here and let us know, and we will fix it. Thank you so much for watching and enjoy exploring the pangenome and pan-epigenome data.